So now that we understand what depression is and we've learned one theory or hypothesis of how depression works, we're going to jump into the pharmacology of depression. So what's happening specifically at the neurotransmitter level with depression. So first thing we're going to do is we are going to really want to focus on the monoamine hypothesis of depression. I'm going to have a separate video on this hypothesis, but here I just want to introduce it, and then we're going to go over exactly what the neurotransmitters involved in this hypothesis do normally and in depression. So the monoamine hypothesis of depression states that depression is the result of a functional deficit of their neurotransmitters norepinephrine and or serotonin at specific synapses in the central nervous system. Now, when I'm talking about norepinephrine in class and in, you'll see on this, like in the exams and stuff, you'll usually see it as NE, and serotonin is usually presented as 5-HT. There's a reason. It's actually 5-hydroxytryptamine, but really 5-HT is just easier to write, or we'll call it serotonin. So that's what I'm talking about when you see it. On the right, we've got a diagram showing a presynaptic terminal, postsynaptic spine, and then we've also got some astrocytes, which are involved in cleaning up that synapse of excess neurotransmitters. And so it's just kind of a nice reminder of what's happening at a synapse, because that's what we're really going to talk about, is exactly how these neurotransmitters are interacting here at the synapse. So under normal conditions, a presynaptic action potential will depolarize the presynaptic terminal, which will open our voltage-gated calcium channels. Calcium will influx, which triggers the vesicles to, synapse or merge with the cell wall and release neurotransmitter into the synaptic cleft here. The neurotransmitters will stimulate postsynaptic receptors, which are the purple things, which are either going to be ionotropic, which means they depolarize the postsynaptic membrane by a sodium influx, or hyperpolarizing by chlorine influx, or they'll be metabotropic, which is triggering a downstream signal, transduction cascades. So neurotransmitters in the synaptic cleft are subsequently taken up by presynaptic transporters, so that would be reuptake, or they're taken up by glial transporters on an extrasynaptic astrocyte, which allows the synapse to reset for the next event. So that's kind of cleaning it out so that it can all happen over again. Now, when we're talking about monoamines, you're going to need to know what those are. So they're made up of two main types of neurotransmitters. The first one, simple enough, serotonin. The second one is catecholamines. Again, I'm not great with pronunciation, but I know what they are. I'm showing them to you here. Catecholamines is about right. So catecholamines is made up of epinephrine, norepinephrine, and dopamine. Again, the main one we're talking about with depression is norepinephrine, but all three, epinephrine, norepinephrine, and dopamine, are categorized as catecholamines. Well, serotonin is almost always excitatory. Epinephrine, norepinephrine, and dopamine can be either excitatory or inhibitory. Other things are going on. You should kind of know this. Both of them can be cleared out of the synapse with transporters. They have their own that are specific to that neurotransmitter, and then also MAO. We'll learn more about what that is and its uses and what it does later, but that's going to be something you'll see a lot in this class, is that MAO. So here's the structures of these monoamines. They are neurotransmitters, hormones that share a common single amine functional group. So you can, see that. And serotonin is probably the most well-known of these monoamines. And like I said, the monoamine hypothesis focuses on the role of norepinephrine and serotonin specifically. So let's get into specifically serotonin, or 5-HT. So this is derived from tryptophan in the brain. So serotonin cannot actually pass the blood-brain barrier, so it just sticks around in the brain because it can't get out. It is a major neurotransmitter in the gut responsible for contraction as well. So how is serotonin synthesized and catabolized? So you're not going to need to know the chemical structures and how things change on the chemical structure, but you will need to know kind of this flow chart of how serotonin is created and how it's broken down. So this image on the right, you're going to really want to know. So we're just going to walk right through it, and it might seem like I'm just reading it off, but it's very important, so I need you to really understand what's happening here. So what we have first is, it's called a dietary L-tryptophan, or just L-tryptophan. That's transported into the brain, so that can actually cross the blood-brain barrier, so that's what goes into the brain, and then it's converted to serotonin via a couple steps. So what's happening is that we have L-tryptophan 5-monooxygenase tryptophan hydroxylase, or TPH. So really we'll just call it tryptophan hydroxylase. Okay, so just this bottom word down here, that's what we're going to call it. And what that does is that's the rate-limiting enzyme, okay, that's going to be involved in the making of serotonin. So tryptophan hydroxylase is regulated by activity-dependent phosphorylation, and it also has a strict requirement for oxygen. So we've seen that rats breathing 100% of O2 will actually increase their serotonin synthesis. So it's a possible effect of once popular oxygen bars, perhaps. And that's, people used to, I don't know if you guys knew this, but people go to oxygen bars, pretty much just inhale a bunch of straight O2, and they got this kind of high feeling. They felt real good, obviously, because of that increase in serotonin. So that kind of makes sense. So that's an increase in oxygen increases your tryptophan hydroxylase, which will then increase the amount of serotonin. Okay, so from tryptophan hydroxylase, it helps create this 5-hydroxy-L-tryptophan, or 5-HTP. This is pretty much just TPH plus lots of oxygen. And then you've got that phosphorylation happening, which then creates serotonin. So that's how we get serotonin. So what I really want you to know here is the L-tryptophan, tryptophan hydroxylase, tryptophan hydroxylase really likes oxygen, and then that phosphorylation creates serotonin. Okay? And then how is serotonin broken down? So it's broken down by monoamine oxidase. So that's that MAO thing that I was talking about just a couple slides ago, where we see that monoamines get broken down from the synapse by MAO. So obviously monoamine oxidase, that's really what it's doing. And so after MAO acts, then you end up with 5-hydroxyindolactic acid, or 5-HIAA. Okay? Now, this final product, you don't need to know quite so much, but I really want you to understand monoamine oxidase, and that is what breaks down serotonin. Okay, so that's the big one. So the big things you need to know here is L-tryptophan, tryptophan hydroxylase, likes oxygen, you get serotonin. Serotonin is broken down by MAO. There we go. And so what are the broad behavioral effects? So serotonin is commonly thought to contribute to happiness, mood, and spirituality. So again, that's why at those oxygen bars, people would increase serotonin and feel happier. So most antidepressants do actually target some sort of serotonergic system to improve mood. So serotonergic system just means a system that has serotonin involved. So serotonin receptors are responsible for the effects of hallucinogenic drugs. So LSD and other hallucinogenic drugs interact, and they activate those serotonin receptors without actually having serotonin. There can actually be a very diverse effect on behavior due to too much serotonin. There can be mood, some migraines, anxiety, attention, inertness, things like that. And serotonin binding capacity has actually been suggested to correlate with tendencies towards spirituality. Of course, this is very questionable. There's been some studies into it, and it's very debatable whether or not spirituality has to do with serotonin, or really what that has to do with it. And most of these studies, they're really hard to reproduce, so not very good studies. Okay. So tryptophan bioavailability. So tryptophan, where do you get it pretty much? So tryptophan is dietary in source, and the availability of having tryptophan can really affect your serotonin production. So if your diet doesn't involve tryptophan, that can actually affect your mood because your body might not be able to produce enough serotonin. So tryptophan is permeable through the blood-brain barrier through specific amino acid transporters, and these are general purpose transporters for large neutral amino acids, and they're very competitive. So just because you eat food with tryptophan in it doesn't mean the tryptophan's gonna, be first in line for that train into the brain. So you've really got to think about that. You can't just, oh, I'm low on tryptophan, I'm going to eat some turkey and get that in. So you've got to kind of, always be eating more tryptophan to make sure that some tryptophan is always getting a ride on the train through the BBB. So consuming high protein meals leads to competition at the large amino acid transporters, and hence decreases tryptophan levels in the brain. whereas eating a meal high in carbohydrates causes branched neutral amino acids to be transported into muscles, which decreases the competition at the blood-brain barrier sites and increases tryptophan transport to the brain, which will then, of course, increase serotonin and melanin, which may actually have drowsy effects. So that's why it's important to have a balanced meal of not just proteins, but also having carbohydrates in there as well to kind of trick some of those amino acids to get out of the line for the brain and move over to the line for the muscles. So here's a fun graph that's showing a tryptophan containing meal or diet versus a tryptophan free mixture diet and how it affects mood. So on the y-axis we have self-rating of sad mood, so the lower on the graph the less sad, higher is more sad. So the red line of tryptophan free mixture is actually showing an increase in a sad mood without tryptophan, and then those that had tryptophan actually experienced less sadness over time, with time in hours on the x-axis. So this was actually done looking at female volunteers with serotonin depletion who are depressed patients in remission caused by an increase in depressed feelings. So this effect can be replicated in healthy volunteers as well to a limited extent, but it does have a pronounced sex bias, predominantly affecting women, which kind of makes sense because depression also does have that sex bias towards females of 2 to 1. So it's kind of cool how just a tryptophan diet can also replicate that. So what happens if there's an inactivation of serotonin signaling? So first is uptake. So the serotonin transporter, which we call SERT or S-E-R-T, or 5-HTT, because 5-HT is serotonin, so then you add a T for transporter. It regulates the synaptic clearance of serotonin. There's 2 polymorphisms of SERT were associated with an increased risk of depression. One of them did fall apart on meta-analysis, but it's still important to note that there were 2. So SERT is actually the primary target of most SSRIs, SNRIs, and TCA depressants, or antidepressants. So many of those drugs target the serotonin transporters. So by these transporters, these are the uptake of extra serotonin in the synapse back to the presynaptic cell. The second one is catabolism by MAO or MAO. So remember, MAO breaks down serotonin, norepinephrine, and dopamine in the brain, as well as serotonin in the gut. So that's just the monoamine one, oxidase. And so you can actually get some antidepressants that target this specifically. So it's that MAOI antidepressants. And so again, it's targeting either increasing or usually decreasing the amount of catabolism by the MAO so that the serotonin can be in the synapse longer, therefore letting the serotonin affect the postsynaptic cell more. So serotonergic projections from the RAF nucleus project to many regions implicated in depression. So the RAF nuclei will project to obviously areas in the limbic system, so the limbic frontal temporal cortex that includes that big BA25 we talked about before. it projects to the thalamus, the basal ganglia, and the hippocampus. So these are all areas in that limbic system that we talked about before being implicated in depression and can really affect mood. So that's where we're going to stop here. Next time I'm going to talk about specifically the monoamine hypothesis and what that says is happening to serotonin and norepinephrine in the brain in depressed patients.